All right, today we are going to finish our notes on the last parts of the main organs that a food source would pass through in the alimentary canal. We've gone through mouth and teeth, where food becomes bolus. That bolus is going to pass through the oropharynx, which is a passageway that air and bolus can share, and eventually moves down into the esophagus. The esophagus superior portion is made of skeletal muscles, which you can control, and then the rest is made of smooth muscles, which you can't. Peristalsis is going to propel that food down into the stomach, where we left off yesterday. There are a couple portions to the stomach. One, the bolus is going to enter through that top sphincter called the lower esophageal sphincter or cardiac sphincter and into the cardiac portion of the stomach. It's going to sit in the body, which is able to expand because of the rugae. And eventually it'll make its way and be digested into chyme when pepsinogen and hydrochloric acid react together to activate pepsin, and chyme will be moved out of the stomach through the pyloric sphincter into the duodenum, which is the first part of the small intestine. Recall that our stomach doesn't absorb any macromolecules. Really, we're just preparing chyme, churning bolus, mixing with enzymes. There's an extra set of oblique muscles along with the longitudinal and circular muscles in the stomach to really make chyme and then pass it on to the small intestine. We really want to move chyme slowly, don't mind my Facebook, through the small intestine to get as much absorption as possible. So let's start off with the small intestine's functions. Please feel free to stop this video and rewind if you need to, but to get all of these notes down and understand them. So the first function is that most of the secretions that are going to come in to the small intestine and break down the last of our macromolecules for absorption are going to come from the pancreas and the liver. We will cover pancreas and liver on a Monday, but these two basically dump right into the first portion of the small intestine, which is called the duodenum. Because of that, we are able to complete digestion or fully break down chyme into the elements, if we go back to that macromolecule sheet, or the raw materials that the small intestine can then absorb. Right now, we really don't have, when chyme leaves the stomach, we don't have the raw materials available to absorb. So this last process will complete in the small intestine. This will allow for the full absorption of the products of digestion. So this is really where we're going to be absorbing our amino acids, which are the small molecules of proteins or building blocks of proteins, where we can absorb monosaccharides, which are the small building blocks of carbohydrates. We can absorb lipids and fatty acids and glycerols, which are the building blocks of lipids. And we can absorb nucleotides, which are the building blocks of nucleic acids. We are also able to transport the remaining residues or waste into the large intestine. Okay, and the large intestine then can be used to remove water and create feces. So all throughout the small intestine, we will still have chyme. Okay, and we'll be removing or absorbing those products of digestion, which are the macromolecules from chyme, and we won't turn it into feces until we start to absorb the water in the large intestine. There are three parts to our small intestine. We can see here, this is our stomach. Remember this portion that narrows out, that would be a sphincter here, is our pyloric region, and immediately the first portion is the duodenum. Now this is where our liver and our pancreas are going to basically secrete enzymes in, and we can complete digestion. Our next portion is all this middle, which is called our jejunum. You can see it's connected by all this connective tissue. It's called mesentery. And there's lots of blood vessels in here. So there's lots of blood vessels within that mesentery that holds the jejunum together. The last portion of the jejunum that kind of straightens out, it's not as coiled, is called the ileum, and that's what's going to open up into the first part of our large intestine. Okay. How this stays into our intestinal or our abdominal cavity, if we think posterior abdominal cavity, so if we touch your stomach muscles, your abdominal muscles, the posterior muscles, or how it's going to attach to that inside wall, is going to be by this mesentery. So all of this that we're looking at here not only holds all of that jejunum, small intestine together, supplies it with blood. So how are we going to get blood there? But also, how are we going to get all these absorbed materials back into the blood so we can use them? So that's why we need lots of blood vessels in the jejunum for that absorption. For absorbing materials, they have to go somewhere. We have to transport them. We'll talk about that in a minute. If we look closely at a microscope image of the wall of the small intestine, this is the mucosa, and we have an image of this that I passed out on a handout. We can see that these are arranged to increase surface area, and surface area is a big concept on the test. Basically, anytime we increase surface area, we are increasing the amount of space that we're allowing the small intestine to do work or absorb. Each one of these is called a villus. The plural is called villi. So many villus are called villi. They're lined with finger-like intestinal villi, many villi. And this is to increase surface area. It's like trying to dry off with a towel, which has those increased surface area because of the thread count on the top, or a flat sheet. We'll be able to do more work because there's more surface area with a towel. Same here. We have more villi. We can do more absorption. If we look closely at each singular villus down below at this picture, we can see a network that runs through the center. This should look familiar to you. We've got the green, which we drew out before as a lymph vessel, and we've got veins and arteries, actually venules and arterioles, that are woven together to make a capillary in here. And capillaries are for exchange of materials, and that makes a lot of sense because this is where we're going to be absorbing all of our materials. We're going to be exchanging that through a process of diffusion into the blood and bringing that into the body tissues. That's called a central core, and that central core is holding all those capillaries and lymph capillaries, and together the lymph capillary is called a lacteal. Okay, so just the lymph itself, how it extends up in here, is called a lacteal. And we can see the capillaries are surrounding that lacteal, and this is where we're going to get an exchange of fluids and transport of fluids from the small intestine into body tissues. Eventually, you want to use the sugar and food you eat for energy. Between each villi, so we're looking right here where my arrow is, we don't see the next villi extending up. We have intestinal glands, which are able to secrete things out of goblet cells. And we looked at the structure of goblet cells. Each goblet cell has those microvilli on them. That was posted in a handout. And goblet cells are secreting mucus so that everything can flow smoothly through here. And actually, it doesn't flow in a one direction. It can flow back and forth. And again, we want this process to go really slow to increase absorption. So here we have specialized cells that are able to secrete mucus to hopefully have a smooth transition of chyme through the small intestine, increasing absorption. There are many different secretions of digestive enzymes in the small intestine. The first are sucrase, maltase, and lactase. Anytime this ends in ASE, that's an enzyme. Okay, it's a protein basically that is able to break down a sugar. Now these are all, I would write in here, which I can't write on my screen, breaking down disaccharides into monosaccharides, which we're able to absorb. We're able to absorb monosaccharides. So sucrase is going to break down sucrose into monosaccharides that it can be absorbed. Maltase is an enzyme, ASE, that can break down maltose, which is a sugar. It's a disaccharide that breaks down into monosaccharides that we can absorb. And lactase, ACE, is an enzyme that'll break down lactose into monosaccharides that your body's able to absorb. Second set of enzymes, there you go, break down disaccharides into monosaccharides. You can tell what they break down based on their name. Is peptidase, not to be confused with pepsin, which is the active form of an enzyme that occurs in the stomach. Remember that mixing with hydrochloric acid. Peptidase, now once those proteins come into our small intestine, they've been unwound. They're in a straight line of amino acids, still a protein. They were unwound or unfolded in the stomach. Peptidase can actually break the peptide bonds apart between each amino acid and break them down into their core amino acids, which can be absorbed. This is going to occur in the small intestine. Do not confuse peptidase with pepsin. Know where both of those enzymes occur and what they do. Our next enzyme is intestinal lipase. Lipase reminds me of lipid. Remember, ASE. ASE tells me this is an enzyme that's breaking down lipids. Now, lipids need to be in their fatty acid glycerol form, which we drew on our macromolecule worksheet. And that's exactly what this does. It's going to break down fats into the fatty acid and glycerols. We drew that into the absorbable form. All right. Next up, how does chyme move through the small intestine? It's going to be mixing movements that move chyme, and we're actually going to try and cut chyme into smaller pieces, again increasing surface area, okay, just like chewing. We're going to cut it into smaller pieces so that we have more space to do work or more absorption. We're then going to move chyme back and forth, okay, so we want to give this time to do absorption, and so it's not a one flow direction out. It's back and forth so that the villi can do work by absorbing. Also, we can do mixing with all those enzymes on the previous slide to maximize absorption. The ileocecal, which is the ileum, cecum, okay, the space between the ileum and the cecum, the sphincter that's there, which is a round muscle, is going to control the movement of chyme into the large intestines. So once we cut the chyme and move it back and forth, and it's moved from the duodenum through the jejunum and down to the ileum, it's going to reach the ileocecal sphincter, or the round muscle between the ileum and the cecum part of the large intestine, where it'll move into the large intestine. Now, what is going to be absorbed in the small intestine are all of our macromolecules. So just to break it down again, we're going to tell you the substance, so you may want to make this chart in your notebook, of what is absorbed and where it gets absorbed. So we know all of this is absorbed in the small intestine, but where exactly? And we're going to go to that structure of the villi to say where it's absorbed. First, our monosaccharides are absorbed. How were they formed? Think back to our mouth. Our mouth had salivary amylase that breaks down polysaccharides to disaccharides. And then once the chyme gets to the small intestine, we have those three enzymes. We have sucrase, maltase, and lactase that can break the disaccharides to monosaccharides, and that will be absorbed into the capillary, which it'll come up in a second. We'll put it in there in just a moment. The capillary of the villi. The second one are amino acids. Well, how did we form these amino acids? First of all, in the stomach, we've got the chief cells and the parietal cells releasing pepsinogen and hydrochloric acid, which mix to form a active form of pepsin. Okay, pepsin unfolds proteins. It's still a protein. It's not an amino acid, but that moves into the small intestine. And then peptidase can break the bond between amino acids, and finally we get this simple building block that can be absorbed. Next, our fatty acids have to be broken down into glycerol. Our lipids have to be broken down to fatty acids and glycerol, and that is done completely in the small intestine by lipase. And lastly, we have our nucleotides, which are easily digestible. Okay. Your small intestine does absorb some water and some electrolytes as well, but let's look at what structure it's absorbed into. Okay, as we can see, all of these are absorbed, most of these are absorbed right into that capillary, which is in the center of the villi, except one. And if you look along here, that's our fats and our glycerol. These are going to be taken up by the lacteal, or the lymph, the lymph vessel in the lacteal. One whole page of your test is basically going to be about how do we get these substances into absorbable forms, where does that take place, and finally, where is it absorbed? So this is something you're really going to want to focus on. You may want to rewatch just what I explained on this whole slide of how that occurs. Once all those macromolecules are absorbed and the chyme moves on to the large intestine, we're going to start to absorb water and electrolytes. No more absorption of nutrients, just water and electrolytes. Electrolytes, again, are just ionized, basically ions. Your magnesium, your calcium, your potassium, that sort of thing. We're going to dry out our waste to form feces. So here we're turning chyme into feces. Here we've got our parts. We start with our cecum, ascending, transverse, descending, sigmoid, rectum, anal, or anus. The functions of the large intestine are that it's going to do absorption, but only electrolytes and water. Again, no macromolecules. All the macromolecules have been absorbed. All the macromolecules are absorbed in the small intestine. We're going to have some secretion of mucus along the way. If not, if you know anyone who's over 50, you have to have a routine 1 year colonoscopy every year to check for polyps. And this is where dried fecal matter along the way can get stuck along the curvature of the large intestine. So we want secretion of mucus to help guide feces out through rectum, anus. It's also going to contain important bacteria that's going to help regulate, you've heard of probiotics, regulate your digestion, and it's normal flora. This is good bacteria. This bacteria is able to make vitamins, and it's going to use the cellulose or sugar in cell walls of plants to do that. So think of like corn, broccoli, that sort of thing. When we first leave the small intestine into the cecum, you see a vermiform appendix there. It's a worm-like, that's what vermiform means, tube that averages about 4 inches long, hanging right by the cecum. It does contain lymphatic tissue, and a lot of people don't know the function of it, or think that there maybe was a function in our ancestral past, but they say there's no function for it or use for it now. Some people don't have it because they've had appendicitis, which is inflammation of the appendix, which it needs to be removed before it bursts. But it's believed that the lymphatic tissue in there is going to encourage some kind of beneficial bacteria that's going to help you regulate vitamins. Your feces then is composed of, once we dry out the chyme, we've already absorbed all the macromolecules in the small intestine. It's dried chyme basically, and it's what your waste is. It's undigested material from your food that your body cannot use or cannot break down. It's going to have some water based on the state of your body, how dehydrated you are, if you're sick, that sort of thing. It's going to contain some electrolytes, again, ions. It's going to contain mucus that's been secreted along its passageway from the ascending, transverse, descending sigmoid colon. And it's going to contain bacteria, specifically E. coli bacteria. And it's going to contain dead epithelial cells or sloughed off cells that have been carried through its journey from the small intestine. And it's approximated that 1/4 of your feces is made up of dead cells from your small intestine and large intestine. The color of your feces and your odor of your feces is due to how bacteria is basically feeding off of your waste. Okay, any undigested material back up here still has to be broken down. Not that your body's going to use that broken down material, but how it breaks down is going to determine your feces color and odor. So you can't just have, just because you can't digest a certain food, you can't absorb its macromolecules, doesn't mean you just goes through your body. You still have to break it down, and the bacteria in your body helps to break it down, and you'll get the color and odor of your feces due to breaking down of that waste, undigested material. Eventually, it's going to hit the last portion of your large intestine, which is an 8-inch chamber that kind of holds, okay, undigested material. It's going to connect your colon or your large intestine to your anus, and we'll specifically say what anus is. So it's kind of hanging on to material there and compacting it before it's out. Now, 8 inches is a lot longer than what you think. So this photo looks like the rectum is small, but it's actually kind of large. It's the last portion. It's a straight portion. It's a chamber, kind of holds on to that fecal matter waste that's going to connect to your anal canal or anus. Okay, the rectum is going to receive stool or feces from the colon, and it's going to kind of signal to you that stool is there. You're going to sense that it's there, and it needs to be excreted or evacuated. You are able, when feces is in the, or stool is in the rectum, to hold it there, but once it moves into your anus, there's 2 sphincters that you may not be able to. So we'll talk about that next. Okay, your anus is then this last 2 inches of the canal. It contains 2 sphincters, so it is actually the opening that excretes. You have an internal and external sphincter muscles. Again, sphincter means round muscle that can control, squeeze off, so nothing can be released or relax, open up, and control the release. The first, the internal sphincter, you don't have control over. Okay. So the second, external sphincter, you do. And so this internal sphincter will open up when the rectum is full. And so that is when it is your clue to relax the muscles of the external sphincter, and you control the release of the feces out of the anus. Okay. We will pick up with the pancreas here on Monday, but we have now finished the 8 organs that food and food material passes through to be digested and absorbed. Please see me if you have any questions, and your ID test will take place on Tuesday, so make sure you maybe want to go to your ID packet or pick up a blank one to start working through memorizing those images.